Hello students, now we will see about new topic that is about lipids. Lipids are defined as esters of fatty acid and glycerols. So here ester we are using the term, it means the bond between oxygen and carbon. We also defined amino acids as methylated derivatives or methane derivatives or substituted methanes, and carbohydrates as hydrates of carbons. Likewise, we are defining lipids as esters of fatty acids and glycerols. These lipids basically basing on size are considered as macrobiomolecules. But basing on weight are considered as microbiomolecules, because weight of lipid molecule is 800 to 1000 Daltons. But normally a macrobiomolecule will be having a weight of nearly 10,000 Daltons. These lipids are classified into three categories. First one, simple lipids. Next one, conjugated lipids. Third one, lipid derivatives. We use a term lipid derivatives in the sense from which or what type of raw material are these lipids formed or synthesized. Derivatives. Simple lipids are again classified into true lipids and waxes. True lipids and waxes. Here, true lipids in the sense that particular lipid is made up of fatty acid plus glycerol only. If glycerol is not there, where any other type of functional group or alcohol is present, then such type of lipids are called as waxes. So waxes are a combination of fatty acid plus alcohol other than glycerol. These true lipids are also termed as neutral lipids. Next, coming to conjugated lipids. In proteins also we have studied, when the protein part is mixed with non-protein part, then we are giving different terms basing on the non-protein part. Here in the same way, if a lipid is binded with any other non-lipid biomolecule, then again the terms are different. For suppose, here we can take examples like lipid plus phosphate, phospholipids. Next, lipid plus carbohydrate, glycolipids. Lipid plus protein, lipoproteins. Next one, lipid derivatives, means from what type of raw material are these lipids synthesized from. Here examples we can take as sterols, next terpenes. These two examples. Now we will start with first one, that is about true lipids. As I said you, true lipids are a combination of fatty acid plus glycerol. These fatty acids are having one functional group and one carboxylic group. This total combination is chemically written for fatty acids. What about glycerol? Glycerol is written as CH2OH, CHOH and CH2OH. So these two chemicals are bonded with each other. How the bond is formed? This is glycerol. Now we will write this one fatty acids like this for our convenience. Then hydrogen plus hydroxyl group is removed in the form of water. Then forms an ester bond. Then the compound looks like CH2OCr, CHOH, CH2OH. Why, madam, in biology, why do we, what is the necessity? Why do we need to learn about this means? I will tell answer. That is, see, you are learning the sentence and the concept as ester bond, peptide bond, glycosidic bond. If we don't have the basic idea how these bonds are formed, then we cannot remember the concept for longer period. So we need to learn about this. Okay, from this we can extract one bit. Glycerols, which are one of the part of lipids, they are existed in the form of monoglycerides, second one diglycerides, third one triglycerides. In the sense, one glycerol plus one fatty acid. Here diglyceride, again one glyceride or one glycerol only, plus two fatty acids. In the third case, triglycerides also, the number of glycerols is one only, but the fatty acids are 3 in a number. So because of this existence, we need to learn how many fatty acids and how many glycerols are being involved. Because of such types of existence of the fatty molecules in different forms, in previous NCERT and previous practice books, I have seen one bit that what is the typical number of glycerols and fatty acids, what is the number of glycerols and fatty acids in a typical lipid molecule? The answer will be, if one glycerol, one fatty acid is existed, still it is considered as a lipid. Because when we are reading about plasma membrane structure like this, we used to represent glycerol like this. There we are framing out diagrams. And we write fatty acids, one pair in a number. For every glycerol, we used to represent the fatty acids are like this. It doesn't mean that fatty acids, if 2 are in number, then only it is considered as a lipid. No. Though one fatty acid, one glycerol are fused with each other by the formation of one ester bond, still it is considered as a lipid molecule. So, basing on the types of fatty acids, They are again classified into essential fatty acids and non-essential. Likewise here, fatty acids are classified into essential fatty acids next non-essential fatty acids. If you see the examples under essential fatty acids, the examples are linoleic acid, which is of 18 carbon, and alpha linoleic acid, which is of again 18 carbon. And a non-essential fatty acids, some examples means already these are synthesized and existed in the animal body. Here we can take examples like palmitic acid and arachidonic acid and stearic acid also. Arachidonic acid. Now, what is carbon number of palmitic acid? 16. Arachidonic acid is 20 carbon in number. What is difference between linoleic acid and alpha linoleic acid? Linoleic acid is also considered to be having omega-6 fatty acids, or they are also called as omega-6 fatty acids. Means they have double bond at 6th carbon, so they are considered as omega-6 fatty acids. That is double bond at 6th carbon. Next, alpha linoleic acid. It is also called as omega-3 fatty acid. Means it is having double bond at 3rd carbon. What is use of these two types of linoleic and alpha linoleic acids? Use is they increase and maintain fluidity in the membranes of animal cells. And maintain fluidity means moisture condition in membranes of animal cells. Here, arachidonic acid, one use we have to study under arachidonic acid, which is considered as non-essential fatty acid. It supports in reabsorption of sodium ions from kidneys in the form of prostaglandins. Means by stimulating the prostaglandins, they support in a reabsorption of sodium ions. Now, essential fatty acids like these two examples are also considered as polyunsaturated fatty acids also. So other term for them is polyunsaturated fatty acids. If we are using the term unsaturated, then what is meant by saturated fatty acids? Animal body is not having benefits from the saturated fatty acids. They get accumulated beneath the skin and are not that much useful like that of unsaturated fatty acids. So, saturated fatty acids are considered to be having, according to chemistry subject, we say as they have single bonds, and these unsaturated have double bonds like that. Saturated fatty acids accumulate, gets accumulated, we write, beneath the skin. So, not preferred for animal body. Here, what examples can we take under saturated fatty acids that are still existed in human beings who are intaking these saturated fatty acids in the form of diet? Examples can be butter fat and meat fat, etc. Butter fat, meat fat, etc. So, for animal body, saturated fatty acids are not preferred. But unsaturated fatty acids are preferred, and most of the unsaturated fatty acids are not synthesized in the body. So, animal body need to intake those unsaturated fatty acids like linoleic, alpha linoleic, etc. in the form of diet. Because they have both hydrophilic polar end and hydrophobic polar, another non-polar ends. So if we write a structure of the lipid molecule, we can consider this one as glycerol, which is also called as polar head, which is hydrophilic in nature. Fatty acids are considered as non-polar ends, which are considered to be hydrophobic in nature. Means they do not allow water molecules to pass through them. Now, we also use another term instead of lipids. Lipids and oils. When we are usually talking, we used to say instead of oils sometimes lipids and lipids instead of oils. But these two are differentiated basing on the melting point. Oils are having very less melting point. Lipids are having more melting point. Are differentiated basing on melting points. Oils have very less melting points. Next. We also will see one more point regarding saturated and unsaturated fatty acids. Some uses. So, saturated fatty acids seen in animals. Next, they are solid at room temperature. Next, coming to unsaturated fatty acids, they are liquid at room temperature and most of them are existed in the plant body only. Unsaturated fatty acids are present in plant and they are liquid at a room temperature. We also have seen the term like true lipids or neutral lipids. So they are accumulated around the visceral organs and protect visceral organs from external shocks. Neutral lipids or true fatty acids or true lipids protect body organs. So these are important points regarding true lipids. Next we will see about waxes. Waxes, as I have said before, are esters of fatty acid plus alcohols. We know different types of waxes. For example, first wax in animal body, that is ear wax. Ear wax, it is secreted by cerumen glands in ear and they protect tympanic membrane. Secreted by cerumen glands and they protect tympanum or tympanic membrane. Next, other example is sebum. We used to give a term for oil secreted from sebaceous glands. That is oil secreted from, what are sebaceous glands? Oil glands. Next, third example we can take as bee wax. It is secreted by the wax glands that are located in the last abdominal glands of worker bees. Secreted from wax glands at last abdominal segments of worker bees. Next, fourth example we also can see in case of aquatic plants. In case of aquatic plants, though we pour some drops of water, they will not get absorbed. They used to float and they used to again drain off when the leaf is turned. Example we can say Victoria leaf and lotus leaf, etc. Waxy coating on cuticle of leaves, plant leaves. This waxy coating we give a term as cutin, called as cutin. We see in case of Victoria plant and also lotus leaves, etc. Next one, in case of polar regions there are some animals and from that also we get wool wax. Fifth one, wool wax. Why wool is having such a natural wool is having such a nature? Because of presence of some types of waxes called as lanolin, which is also called as wool wax. So these are few examples in case of waxes topic. Next we will see about conjugated lipids. Previously we have seen some of the examples. First example we can write under conjugated lipids as phospholipids. means the lipids that are combined with phosphate molecules. Mostly these are seen in case of nerve membranes. Now, one glycerol, when attached with one phosphate, then we can give a term like lecithin. Glycerol plus phosphate of a lipid combinedly can be termed as lecithin. So, some of the MCQs are there. Lecithin is related to the phospholipid or plasma membrane we used to tell as answer. What does it mean? Means that plasma membrane is having a combination of fatty acids, glycerols plus phosphate. So, we call biphospholipid layer. But the glycerol along with phosphate is termed specially as lecithin. So, lecithin is a part of plasma membrane. Mostly these are seen in nerve membranes. Next, glycolipids also we see in case of plasma membrane. They are meant for protection. Glycolipids, they are protective in nature. Next, when a nitrogen base is combined with a lipid, then the term changes. When nucleotide or nucleic acid is combined with protein, there we have given a term as nucleoproteins. But nitrogen base, it can be adenine, guanine, cytosine, thymine, uracil, anyone. Nitrogen base plus lipid. Then the name of the term changes as sphingolipid. Sphingolipid. And when lipids are binded with proteins, obviously the term will be lipoproteins. So, protein plus a lipid. Lipoproteins. Next topic we can take derived lipids or lipid derivatives. Derived lipids or lipid derivatives. So here there are two examples. One is sterols. Second one, terpenes. Sterols are considered to be derivatives of type of compounds called as phenanthrenes. Are derivatives of phenanthrenes. Terpenes are considered to be derivatives of isoprenes. Phenanthrene is considered to be a 3-ring structure. Because if we know the differences, so we can remember easily. Is a 3 benzene ring-like structures. Ring structure. Isoprenes are considered to be 5 carbon compounds. 5 carbon compounds. These sterols are of plant origin. Again, these are of different types like Stigma Sterol and Phytosterols. Since these are of plant origin, they are also called as Phytosterols. Phytosterols. Now, phytosterols are of two types. One is Stigma Sterol. Stigma sterol is obtained from soya beans and it can be converted artificially, means in vitro, into progesterone from soya beans. Stigma sterol can be converted into progesterone. There are many uses of progesterone in reproductive systems of both male and female. Other types of sterols are, one is ergosterol, which is seen in the plasma membrane of fungus. Other one is zoosterol, which is also called as cholesterol, and it works as an insulator. Its melting point is 147 degrees to 150 degrees centigrade. In plasma membrane, the same cholesterol, it provides strength to the total cell by existing in the plasma membrane. This cholesterol is considered as an precursor of vitamin D. Cholesterol is precursor or raw material that is required for synthesis of vitamin D. Vitamin D. Next, other examples are bile acids. Bile is the secretion from gallbladder or from the liver, we commonly say, that is considered as emulsifier. Emulsifier means, or emulsification means, conversion of large particles of lipids into very small particles such that enzymes can easily act on them. Bile exists in the form of bile salts, bile acids, and also bile pigments. So now we are reading here about bile acids. What are bile acids? What are they used for? Cholic acid. Next, deoxycholic acid. Next, chenodeoxycholic acid. These three are considered to be bile acids. They are helpful in absorption of fat-soluble vitamins back to the body. And also, these type of bile acids, they maintain metabolic homeostasis, in the sense, when metabolism means breakdown of any particle, carbohydrates or proteins or lipids in the form of amphibolic pathway has occurred, some energy is released. Energy releasing means some heat is generated. When that heat is generated, then body becomes overheated. That is controlled and regulated by these bile acids. So bile acids, they help in absorption of fat soluble vitamins. Next, and maintain metabolic homeostasis. Next, another example we can take, some of the these sterol types of substances are also produced in the form of hormones or existed in the form of hormones that we call as steroid hormones. So sterols in form of steroid hormones. In human body, we have two examples under steroid hormones. Many are there, estrogen, progesterone, etc. Here we will take just two examples and we will learn. Progesterone, first of all, if we take progesterone, it is secreted by many organs in the body. For suppose ovaries produce progesterone, corpus luteum produces progesterone. So what is use of this one? This progesterone is used to maintain the pregnancy throughout the pregnancy period and also to regulate the menstrual cycle. So it is used to maintain pregnancy and regulate menstrual cycle. Another example, testosterone will take, or estrogen also we can take, which are majorly responsible for the follicle formation in females. So we can take estrogen as an example. Major function in female reproductive system or in development of female secondary sexual characters. Female body. So these are few examples of sterols. Now, what is use of these terpenes? These are used as during the synthesis of vitamin A. These are used, and also these are used for synthesis of carotenoids, and also they are considered to be very important structures or the substances for providing good flavor and aroma, etc. So they are considered to be existed in eucalyptus oil, some camphor, etc. These terpenes are existed in a natural rubbers in the form of polyterpenes, are existed in natural rubbers in form of polyterpenes. These are used during vitamin A synthesis. Next, used during carotenoid synthesis. Carotenoids are considered to be the precursors of vitamin A synthesis. Next, they play very important role in the odor that is released from camphor eucalyptus oil. They are responsible for odor in camphor and eucalyptus oil. So this is about lipids. So in biomolecules, we have finished about carbohydrates, proteins and lipids. The next topic will be nucleic acids, and after that we will see about enzymes, so that this chapter will be ending.